Hello, good afternoon, everyone. I know you guys just finished your math quiz this morning.
Mhm.
Hopefully you still have a bit of energy left to do the lesson. And of course, you still have to do the quiz for this course today, right? You will have access to it until 11:59 p.m. tonight. Um, so I will keep the lesson short. We'll skip the break and we'll go all the way to 2 o'clock. And that way you can still have some time to study if you need to, uh, and uh, and do the quiz. So we will start on lecture 3 today. Lecture 3 is about cells. One of the first thing that you learn is the characteristics of living things, right? And one of the characteristics is the organization that is associated with life. So not only is the organism showing complexity as a whole, but that level of organization exists all the way at the molecular level, right? So we know that the basic building blocks of life are atoms. Okay, in fact, anything that exists in the world is composed of atoms, right? But in terms of living things, we have carbon, hydrogen, oxygen, nitrogen, phosphorus. These are the main ones that makes up 96% of the human bodies. Now, these atoms can come together to form molecules, right? Such as H2O, right? C6H12O6. You might even remember seeing this picture, right? It's something that we did in lecture 1. So these molecules are quite small. Water has 3 atoms in it. Glucose is a little bit bigger. It has 24 atoms, but still relatively small. Then we have the macromolecules, the big ones that we've learned, right? The carbohydrates, the proteins, lipids, and nucleic acid, right? Nucleic acids. Acid. So these are big molecules. They have hundreds and thousands of atoms that compose of them. So that's where we left off in lecture 2. Now today we're gonna move up the ladder one more level, right, to the cellular level. Right? So macromolecules can come together to form organelles, which are tiny organs that is found inside a cell, and it helps them perform specific functions. And in fact, from now on, between lecture 3 all the way to number 9, I would say, we will be learning about the cell, different aspects of the cell. Right? Today, we're just learning about the structure of a cell and what's in it. Right? Later on, we're going to learn more about the processes that are taking place within the cell. Right? And then lecture 10, we'll learn about tissue. And then next semester, we will learn about the organs and organ system. So that's where we're heading. Now, how big is a cell? If you think about a human, like an average adult would be about 1.5 1.6 meter tall, right? Anywhere from 5 feet to 6 feet, that's like the average height. But if you take that and you divide it by 1,000,000 times, then you would be talking about the size of a cell. Okay, so 1 meter has 10 to the 6 micrometer, right? So a millionth of a meter is a micrometer. And animal cells, plant cells, they are in the range of about 10 to 100 micrometer. You cannot see these things with your naked eye, right? You will need to use a light microscope. Smaller things exist, right? The macromolecules that we talk about, the proteins, the lipids, they are close to the nanometer range. Right? 1 meter has 10 to the 9 nanometers. Okay, so a billionth of a meter is how big 1 nanometer is. So these, you can't even see them with light microscope. You need very powerful electron microscope to be able to see them. All right. So what is a cell? Cell is defined as the smallest unit that still has the properties of life. You are familiar with the characteristics or properties of life, right? They are able to maintain homeostasis, they respond to stimulus, they will grow and develop, so on and so forth. So all these properties, what is the smallest unit that still retain all these properties? Well, it's a cell. Okay? If you go any deeper into the cell, right? If you look at protein, which is smaller than the cell, right? Protein is no longer defined as alive, right? Because proteins is just on its own, doesn't require energy, it's not gonna reproduce, right? It's not gonna be able to maintain homeostasis, right? So protein is no longer life. So the smallest unit of life is a cell. Now we have three pictures here. Anybody? These are different cells. Anybody want to tell me what this might be? This you probably seen this before, like in movies, right? You know, on the internet maybe. What kind of cells do you think this is in the body? Hmm? Any guess? Brain cells. That's right. Right? These are neurons. Right? They are found inside our brain, and they're making connections with other neurons, right? We have the streptococci here, which can give people strep throat, for example. And then here we have something called amoeba, okay? Amoeba. They live in fresh pond water, but sometimes they make their way into like swimming pools and stuff, or like if people swim in the lake and you know they could pick up the amoeba, and it could cause them to become ill. Okay, they can get like diarrhea and stuff like that. So the cells, right? Three different kinds of cells. They all look very different. Um, but there are some features that are shared between cells. So here are three features that are common to all cells. The first one is that they all have a cell membrane. Okay? They all have a cell membrane that separates the living environment on the inside from the non-living surrounding. The cell membrane is also known as the plasma membrane. Okay? It's important for controlling what goes into the cell and what leaves the cell. After all, you don't want things to be moving in and out without regulation, right? You might be losing valuable nutrients and you might be letting harmful things to come into the cell, right? So there is some kind of gatekeeping going on. That's the job of cell membrane. So regardless of what kind of cell it is, right, it will have to have a cell membrane. Number two, it has to have genetic material, right? So we know genetic material can exist in the form of DNA, right? Sometimes it can exist in the form of RNA. And the genetic information basically programs the cell, right? It allows the cell to behave in a way that it should. And lastly, we have something called the cytoplasm. Cytoplasm is kind of like a gel-like liquid that is inside the cell. It makes up a majority of the volume of the cell. Yeah? And you have all these structures that are kind of suspended inside this cytoplasm. All right. So we have these things called the organelles that are in the cytoplasm. Sometimes they would have a nucleus to package the genetic material, but not all cells have a nucleus. Okay. Right? Only some cells have a nucleus. All right. 1 second. All right, so those are the three common features of all cells: cell membrane, genetic material, and cytoplasm. Now there is something called a cell theory, which actually has 4 different parts to it. The first part goes like this: every living thing is made up of one or more cells. Right? So we have a single cellular organism here, called the paramecium, is just one cell. The amoeba that we saw earlier, that's also just one cell. Right? So these are unicellular. You'll hear me bring the paramecium back a few times as we progress. There is something called C. elegans, which is 3,000 cells. Okay, so C. elegans is this worm, microscopic worm that scientists actually use to test a lot of things. So most recently, they were able to mutate one gene. A gene is a segment of DNA. Okay, they're able to change the segment of the DNA in the worm, in the C. elegans, and it causes it to double its lifespan. Right, so that's very exciting because humans also have that gene. Right, so maybe we could study the effects of the gene on aging, right, and ultimately perhaps increase lifespan. Humans are much bigger. Right? We have 37.2 trillion cells, right? Plus or minus, depend on like how big you are, right? So these are multicellular organisms. Okay, so if it's an organism, it's going to make up of one cell or many cells. Now, even though the cells are part of our body, right? But they are individually alive. What that means is you can actually take cells out of your body and grow them in a culture. Okay, so in a petri dish. So these little plastic containers, they're called petri dish, and the red stuff is a nutrient solution. So you can supply the cells with nutrients, and they would be quite happy. They would grow, they would divide, but not forever. Okay, they would grow and divide and maybe live for a couple weeks, and then they would die. Okay, but still, they are able to survive without the whole organism, right? Okay, but you got to give them the right condition. Some cells can grow forever, even out of the organism. Okay, normal cells, they would die after a while, but some cells, like cancer cells, they would be able to grow forever. So there are these things called cell lines. Cell lines. And people can buy cell lines from biotech company or from biotech companies. Okay, and they would allow scientists to do research and experiments on them. Okay, so there are different kind of cell lines. These are cells that would grow on forever. Okay, they have engineered it to grow forever, or sometimes they are taken from cancer source, and so they're able to live forever. We call them immortalized cell lines. Okay, so people can do drug testing on them. They could make vaccines with them. They could study the behavior of mutations, right, using these cell lines, and they are quite valuable as a research tool. Yeah, like if people want to test the effectiveness of a new drug, they're not going to test it on animals right away. Okay, even with animal testing, there are ethical guidelines, right? Okay, so like you must show that there are promises in theory, right, before you can move on to animal. And to show that it works in theory, you would use these cell lines. Okay, so there is a famous cell line called HeLa cells. They're very common. You can even buy it online. Okay, you can get it shipped to your home, but then you wouldn't be able to grow them, right? Because you don't have the facility to do so. Yeah, but in the lab, it's easy to grow them. So it's taken from this lady. Her name was Henrietta Lacks. That's why it's called HeLa cells. She had ovarian cancer, and the doctor took a biopsy of her tumor. Yeah, a biopsy is like a sample, right? And then he grew them in the petri dish, in the culture, to see what will happen. And to his surprise, they just keep on going. They just don't die. Okay, and later on, I think it was maybe sold to a pharmaceutical company or something, or like a biotech company rather, nowadays you can buy the HeLa cells for research, right? So the family of Henrietta was not happy about this decision because they say the company did not have the consent of the person to be profiting off her cells, right? Essentially. And so they file a lawsuit against the company. The company is Thermo Fisher's, and the company tried to push back and discredit their lawsuit. So there is a few back and forth, but eventually, I think the judge awarded the family some money. They think they are in the right, and that the company actually must pay them for unauthorized use of the cell, basically. So we're talking about the different parts of the cell theory, right? All cells are made up of one or more cells. All organisms are made up of one or more cells. Number two, the cells are individually alive. You can grow them outside of the body. Number three, all living cells come from division of pre-existing cell. Nobody knows where the first cell came from, but we know cells can split and make more cells. Okay, so this original cell is called the parent cell. And then these ones that you get afterwards, those are the daughter cells. Okay, and this process of making a copy of itself is called mitosis. We'll learn more about mitosis in lecture 7. Okay. So you guys told me that we have 46 chromosomes, right? So in the process of mitosis, when we split the cells, you will have to duplicate the chromosomes, and then each of the daughter cells are going to have the same number of genetic material. So that's the fourth part of the cell theory. Cells containing hereditary materials will be passed on during the division. Okay, so this is onion cells that you're looking at, and the cell is actually splitting right now. It's dividing. All these red stuff that you're looking at, those are the genetic material. So that's the cell theory. Now we cannot see the cells; they're too small with our eyes, right? We have to use microscope. So the first microscope that was documented was created by this person, okay? Antony van Leeuwenhoek. Right? He created this handheld device, and he put some pond water at the lens and at one end of the lens, and then he just was observing it, right? And he noticed there is an entire world that was unseen before that exists in a few drops of pond water. He saw things that was moving around, and those turns out to be single cell organisms, right? Like the amoeba and the paramecium that we mentioned about. Nowadays, light microscope are very, very common. Right? I think I told you we have some in the lab at school. They're pretty cheap, like $120 each for a microscope, and it's relatively decent. But for very high power resolution, if you want to magnify things a lot, then you have to use electron microscope. Okay? Electron microscope are very expensive. Okay, sometimes they can cost as much as a luxury car. All right, and there are two kinds here. We have the transmission electron microscope, and then we have the scanning electron microscope. This is good for looking at 3D view of your specimen. You can even observe them while they're alive. This one, you would do a cross section. You would cut your specimen, and you will study the structures that are inside, right? So the thing would have to be dead. Right? Does anybody want to guess what we're observing here? What are we looking at? It's something that's found in the body. Anyone know? No guesses? All right, so these are called cilia. Cilia. which are found in airway. Okay, so when we breathe in like pollutants or some kind of dust particle from the air, the cilia in the airway, they will sweep these particles up and then you can spit it out, right? People who smoke a lot, they damage the cilia, so there is no way for them to naturally remove the trapped particle. They have to physically cough it out, right? So a lot of smokers, they have what's called a smoker's cough, and that is as a result of damaging their cilia from smoking. All right, let's take a look at this question. We will take it up in a bit. All right, I'm gonna launch the poll. Okay, not bad. Everybody who answered got it right. How about this next one? Okay, the correct answer is actually nucleus. Nucleus is not present in all the cells. Let's go back and take a look here. There are only three things that are a common feature of cells: cell membrane, genetic material, and cytoplasm. So let's go back here, right? Cell membrane or plasma membrane, cytoplasm, and genetic material. So C is the exception. Okay. Yeah, I know this one is a little bit tricky. A lot of people get it wrong, but that's okay. Now you know the answer. Hopefully you'll be fine. All right, let's go on and see. There are two kinds of cells. Okay, we can categorize it as a eukaryotic cell, or a prokaryotic cell. Okay, so that's like the adjective, but the noun is eukaryotes. Okay, Y-O-T-E, and prokaryotes, Y-O-T-E. You can use them interchangeably. Eukaryotes are the more complex cell. They usually are bigger. They have a lot of stuff in them, a lot of organelles in them. And the key distinction between the two is that this is the one that has a nucleus. Okay, the eukaryotic cell has a nucleus in them. And the nucleus is where you will find the DNA. Okay, contains genetic material. Okay. So animal cells are eukaryotic cells. Plant cells are eukaryotic cells. Okay, so in our body, if you take a look at the liver cell, if you take a look at you know, lung cells, brain cells, heart cells, they are all going to be eukaryotic cells. Okay. On the other hand, prokaryotic cells are smaller, simpler. Okay. They do not have a nucleus. The DNA just kind of hangs around in the middle. There's no structure to contain them. Okay? They don't have a lot of organelles, just a few of them. And most bacteria are prokaryotic cells. Okay? Does anybody know the name of any bacteria that you've heard on the news, that you read about? Okay, bacteria name usually has a single letter followed by another word. Okay? You must have heard some of them, right? Anybody? Help me out here. That's right, E. coli. Very good, right? E. coli, right? So people heard of E. coli contamination, right? You might have that on like the romaine lettuce or something like that, right? Any more? Just wondering.
Salmonella.
That's right, salmonella. That's another one, right? There could be salmonella contamination as well, right? In various products, produce, for example, right? Cold cuts. Okay, so these are prokaryotic cells. They are simple. They don't have a nucleus. Listeria, excellence. That's another one, right? That causes listeriosis, right? So all these are prokaryotic cells, right? They are simple. They don't have a lot of structures in them, but don't let them fool you. Just because they're simple doesn't mean they're not harmful, right? All these things that we talked about, E. coli, listeria, salmonella, streptococcus, right? All these things can kill, right, a person, right, if they're infected with it. So all these cells, regardless if they're eukaryotic or prokaryotic, they are gonna have a cell membrane that protects them, that surrounds them. Okay, and the cell membrane function as a selectively permeable barrier. Right? Permeable means it will allow things to pass through. So it's not like a brick wall. Okay, like it's permeable. It will let something pass through, but not all things. That's why it's selectively. Okay, selectively permeable. And what is the cell membrane made up of? Well, you better know this because it's on your quiz today, right? It's made up of the phospholipid, right? But it actually is not just one layer of phospholipid. There are two layers of phospholipid. That is why we call it the phospholipid bilayer. Okay, phospholipid bilayer. It means there are two layers of this stuff. And the way that it is being arranged is that you will have the hydrophobic tail that is sandwiched between the hydrophilic head. If you think about a cell, okay, this is the inside, it's mostly water. The outside environment is also mostly water. So it makes sense for the hydrophilic heads to be facing outside or inside, but you're gonna have the hydrophobic tails that are sandwiched in between to create that barrier. Okay. It turns out the cell membrane is not only on the surface of the cell. I mean, when it's on the surface, then it's called cell membrane. But then there are organelles, there are structure inside the cells that also has membrane surrounding them. Okay, some even have two membranes surrounding them, and we're gonna look at that probably next class. This is an picture taken from an electron microscope, very high power. And you can see the two thick black lines here. Those are the hydrophilic heads. And then the space in between, that's the hydrophobic tails. Okay, and that's the cell membrane essentially. This is the outside of the cell. And then this is the cytoplasm, the inside, cytoplasm. And right here, that's the cell membrane. Okay. Hopefully that makes sense. Now I'm gonna encourage you to take out a piece of paper and then we're gonna draw stuff. All right. Because that this next part, if I just read the slides to you and show you stuff on the slide, like it's really hard for you to remember. Okay. Instead of doing that, I'm gonna build a cell with you, and then we'll go back to the PowerPoint slides. Okay. So take out a piece of paper. And we're gonna use the whole thing. Okay, we'll add more stuff to it as the lecture goes on. Okay, but like don't do a tiny diagram here. Okay, my diagram is gonna be really, really big. Okay, so use up the whole page. Right? I'm gonna draw a circle, and your circle should be like, you know, this big on your piece of paper. Right? So get a piece of paper, get some highlighters if you have, markers, pencil crayons, that kind of stuff. Just gotta give you a moment to get ready, okay? Okay, so please draw along, all right? Don't just watch me do it. So we're gonna look at eukaryotic cell, which is us, right? Our cells are eukaryotic. So there's gonna be a nucleus right here. This big circle is called the nucleus. Okay, and you will find nucleus in eukaryotic cells, but not prokaryotic cells, not bacteria. Okay, you'll find it in human cells and plant cells. Okay, now this nucleus is going to be surrounded by a membrane, but the membrane doesn't wrap around the whole thing. It has a little bit of gaps in it. Some gaps. All right. Now I cut open this nucleus so we can see the inside, but imagine this is covering the whole thing. All right, but I'm not going to shade it in. I'm going to open it up so we can see the inside, right? So this purple thing that's wrapping around the nucleus is called the nuclear envelope. Okay, nuclear envelope, which surrounds the nucleus. Okay. But then there are these gaps, right? These openings, these gaps in the envelope. We call them nuclear pore. Okay. One pore, many pores. Okay. And the function of a nuclear pores is to allow things to enter or leave the nucleus. Okay, so certain things will need to come out, certain things will need to go in. If you wrap it all up with the envelope with no gaps, then you can't really go through it, right? So you gotta leave some gaps. Is it okay? So, inside the nucleus, we have another structure. That is called the nucleolus. And the nucleolus makes something called ribosome. We'll talk about that in a second. Just write it down for now. Makes ribosomes. So if you're having a hard time visualizing this, think of avocado, okay? The seed is like the nucleolus, okay? And then the skin is like the nuclear envelope, right? It surrounds the whole thing. And now imagine taking a fork and then poking holes on the surface, right? So like you would have some holes here, holes, right? Those holes would represent the nuclear pores. Are we together on this? If you're lost, ask something, because it's only going to get more complicated. Anything you need me to clarify? Really? You guys all got that? That's great. Okay. So what else is in the nucleus? Well, in the nucleus, you're going to find these genetic material, right, in the form of chromatin. Chromatin. Right? Now, I can hear some of you screaming at the screen. What do you mean chromatin? Isn't it DNA? Isn't it genetic material DNA? Why is it called chromatin all of a sudden, right? Well, DNA is very, very long, the double helix, right? Very long, and if you try to fit all the DNA into your nucleus, it won't fit. It's too much of it, okay? And even if you manage to stuff it in, they will get all tangled up. Think of like sewing threads, right? Okay, like think of the sewing threads that you have. Like if you unwound the whole thing, it's just gonna get into like a tangle mess, right? So that's why the sewing thread, we're gonna put them around spools, right? Like this. And then you can pack it nicely in your sewing kit. So the red thread's not gonna tangle with the green ones or the blue ones and so on and so forth, right? So that's what happened here. We have the DNA, which then wrap themselves around proteins, okay? These circle balls, they are proteins, okay? Kind of like threads around spools, right? And we get our chromatin. So essentially, these are DNA wrapped around proteins, right? To package them nicely so that they are going to fit in the nucleus. They're not going to get tangled. They're not going to break. Okay. But then there is another C word that makes this confusing, right? Like you hear people say chromosome. So what's the relationship between chromatin and chromosome? Well, it turns out when a cell needs to divide, you need to move the genetic material around. Okay, you need to move it to different cells, for example, right? And chromatin, even though they're compact, but they're still not very good for moving around. Okay, so you need to package them more. Okay, so the chromatin, when you need to move them, it's gonna condense even more. Okay, and that is when you get your chromosome, and that's what chromosome looks like, right? Usually when you see them on like TV or pictures. Okay. So just like us, right? Like if we have to move to another place, move to another apartment or something, we gotta pack our stuff before we move, right? And once we finish moving, we can unpack them. Same situation. The chromatin are the normal state when the cell is not doing when the cell is not dividing, so you don't have to move the genetic material around. But when we need to move them, you need to pack them up to become the chromosome. You move them where they need to be, then you can unpack back into the chromatin state. Is that okay? All right, so all that you will find in the nucleus. So let's draw more. Okay. I am really hoping you guys are drawing with me, okay? Like if you're just watching me do this, then it's not very useful for you, okay? So that nuclear envelope actually extends into the cytoplasm. Okay, it actually forms these tunnels, okay, these networks that are extending into the cytoplasm. Okay, and I'm going to use a different color here to draw more of them. Extra network here. Okay. So these networks, they are called endoplasmic reticulum. Endoplasmic means within the cell. Reticulum is network. Okay, so we have another set of endoplasmic reticulum. Okay. Now, don't worry about what they do at the moment. We just want to know where things are. Okay. So this is an extension of the nuclear envelope. Right? It's connected to it. It just branches outward. Okay. And remember, this is where the cytoplasm is, right? So let's go back here for a second. The nucleolus makes the ribosome, right? Ribosome is another organelle. It's really, really small. And they will leave the nucleus through the nuclear pores. Some of the ribosomes will be floating freely in the cytoplasm. Okay, they'll just be on their own. So these are free floating ribosomes. Okay, just sloshing back and forth on their own within the cytoplasm. In the cytoplasm, the liquid that's inside the cell, right? And over here, some ribosome will attach themselves onto the endoplasmic reticulum. Okay. So when you look at this under the microscope, the ones that have the ribosomes on them, it looks bumpy. So people call them rough endoplasmic reticulum, RER. On the other hand, some endoplasmic reticulum does not have ribosomes on them, so we call them smooth ER. And they have different functions, which we will learn later on. But that's all I wanted to show you at the moment. Okay, I just want you to see where things are at and how they are related structurally, how they're connected. Does anyone have any question before we go back to the PowerPoint slides?
Yes, please. Where is the cytoplasm?
Oh, okay, yeah, I know, right? Sorry, I was not being very clear. So don't draw this next part, okay? But this is the nucleus. This is the whole cell, right? Like I just didn't draw the cell membrane, right? Okay, so everything here that's inside, that's part of the cytoplasm. Does that answer your question? Was it Mina? Mina?
Yes,
Okay.
Mina. So even the like chromosome and all of that, that red part for the
Oh no, sorry. This part is
that
not in the cytoplasm. This part is all inside the cell here, right? I'm just drawing a diagram to show you the meaning of chromatin.
Oh, okay.
Yes,
Okay.
sorry about that.
That's okay. I just wanted to like visualize
Yeah, so like
it.
if I have to draw the cell, right? Like I guess it's like this. Is that okay?
Yeah. Okay. So okay. All right. And then the chromatin is made up of the chromosome and DNA and all of that.
That's what chromatin is, the DNA that's wrapped around the proteins. And then if you need to move them around, it would further condenses to form the chromosome. Okay?
Sorry, can you repeat? I'm so sorry. Can you
Sure. No
repeat that?
worries. Okay. I'm sure other people have the same question. Look, so this is a regular cell with a nucleus, right? And then you would have your genetic material on the inside. So those genetic materials are packaged in the form of chromatin. Okay, they are packaged, but they are not super condensed. Okay, like you can pack them even more, but we just don't need to do it at the moment. The only time you need to pack it even more to become the chromosome is when you need to move the genetic material around. Okay, so the next question is, when do we need to move them around, right? When the cell divides. Okay, when you divide the cells, you need to make sure to separate the chromosome so that each of the daughter cells are going to have the same number, right? So to make it easier to move them around, they condense further to become the chromosome. Does that answer your question?
Yes.
Okay,
And then when they split, that's mitosis when the
that's
cells
right.
split.
And then after they split, after you finish moving the chromosome, then they could unwound, they could decondense back into chromatin, right? because now we don't have to move them anymore until the next time.
Right. Okay. Okay. I got it. Yeah.
Great. Good question. Anyone else? I know you guys are like super busy day, right? With the bio test and the bio quiz and math quiz, and your mind is probably not fully here. So I do appreciate you guys coming to class. Yep, I will keep it short. Let's go back to the slide for a second. Everything on the slide we've drawn already, right? But let's just look at the professional drawing, right, to see what it looks like. Here we have the nucleus, the purple ball. Okay, and then in the center of it, right, that's where you will have the nucleolus, the seed of the avocado, right, as an analogy. And then you see all these openings that are on the top, right, on the nuclear envelope. Those are the nuclear pores. That's how things enters or leave the nucleus. Okay, one of the things that will leave the nucleus are ribosomes, represented here by the yellow dots. Okay, so the nucleolus makes the ribosome. They leave the nucleus through the pores, and they will attach themselves to the ER, to the endoplasmic reticulum. Some ER will have no ribosomes on them. Without ribosome, they are smooth ER. With the ribosomes, they are rough ER. Some ribosome will just be floating freely on their own. They don't have to attach to anything. Okay, and it's not being shown on this picture. This explains the same thing I was trying to explain earlier with different levels of DNA. Okay, so in the freest form, in the form that is just DNA, not wrapped around anything, that's just the double helix. Okay, but like I said, they won't fit right in the nucleus because you have so much of them. So you need to wrap them around these proteins called histone. Name is not important. And then you get the chromatin. And then when the cell divides, you need to condense it even further, and that's how you get your chromosomes. Right? That's, you know, when people say we have 46 chromosomes, that's what they're referring to. All right, more on that later in the course. So I already told you nucleolus makes the ribosome. Let's take a look at the ribosome. Okay. Ribosomes are really small, right? I just draw the little blue dots, right, on it. So if the nucleus is like a basketball, okay, nucleus is like the size of a basketball, then the ribosomes are like sesame seeds, very small. But if you zoom in and take a look at the ribosome, it actually is made up of two subunits, a large one and a small one. Okay, think of it as a burger, okay, burger bun. You got the top bun that's bigger, and you got the lower bun that's smaller, right? So that's your ribosome there. Usually they're not together, okay? It's only when the ribosome is doing its job will the two subunits come together. Right? So what is ribosome made up of? It's made up of two things, actually. It's made up of proteins and RNA. You might recall the last slide of lecture 2, I showed you there are three kinds of ribosome, three kinds of RNA. There's messenger RNA, which you know you heard of, right, from the vaccine. There is rRNA, right? That's the type of RNA that makes up the ribosome. Okay, so ribosomes are made out of proteins as well as RNA. The function of ribosome is to make proteins. Okay? You should know by now, I hope you know, for the sake of the quiz, that proteins are made up of amino acid. So something has to connect the amino acids together, right? Okay? Like how do you form the peptide bond, right? How do you connect the third one, the fourth one, so on and so forth, right? Well, that is the job of the ribosome. Okay? Ribosome is the one that will be taking the amino acids and forming the peptide bond. All right. Here we have a picture of the endoplasmic reticulum. That's what these channels are. And all these, it's really hard to tell, I guess, but all these black circles, those are the ribosomes. Don't worry, on the quiz, number 2, right? Quiz number 2, okay? I will only give you cartoon pictures. Okay, I'm not gonna give you like the real thing and ask you what it is. That's like, impossible, right? So I'm just gonna show you a cartoon picture and I ask you what it is. And then you say, oh, ribosome. I remember the burger, right? Large subunit, small subunit, ribosome. Okay, so you already know this. I'm just gonna read it to you. Ribosomes are made in the nucleolus and exits into the cytoplasm through the nuclear pores. I must have said that 3 times already. Some ribosomes exist freely in the cytoplasm, just sloshing back and forth, not attached to anything, right? While others are attached to the endoplasmic reticulum. Now, this next slide is important, okay? It sets the stage, the theme of what you will be learning for pretty much the rest of the course. Okay? It's called the central dogma of biology. I have to admit, the first time I see this word, I have no idea what it means, okay? But it's just like a unnecessarily complicated word that means the same thing as theme, okay? So central theme of biology. Right? So you remember I told you the instruction to make protein is encoded within your DNA, right? Your ACTGs, those would make up instruction to make proteins, okay? But the DNA is in the nucleus, and the ribosomes, the one that is supposed to make the protein, is in the cytoplasm, right? So how do we extract the information from it? Well, the DNA is really big. I told you that already. They won't be able to leave the nucleus. Okay, it won't even fit through the pores. And even if it would, okay, we don't want to take the DNA out of the nucleus. Like, what if we damage it, right? And that's not gonna have a good outcome, right, for the cell. So we want the DNA to remain in the nucleus. So when we want to make a protein, we need the instruction from the DNA. So instead of taking the DNA out, we will just make a copy of the DNA. Right? And we can do that by copying the DNA in the form of RNA. This is called transcription. Okay, to transcribe something is to copy, right? And this is the same language. DNA is nucleotide, RNA is also nucleotide. It's the same language. That's why we call it transcription. All right, think of it like this. Let's say at the school library, there is this 800 pages book, okay, that has practice multiple choice question in it to help you with your biology test. All right, so it's a very good book, right? Okay, but they only have 1 copy, and they don't want you to sign it out. Okay, they say no, this is our only copy. I don't want you to, lose the book or damage the book, right? You can only use it at the library, but you really want it, right? So what do? You make photocopies, right? But you are not going to photocopy the entire 800 pages. You're only going to photocopy maybe the topic that you want, right? Okay, maybe 10 pages of it or something like that. Right? And then you take it out of the library and use it at home, right? And that's exactly what's going on here. DNA is like that textbook, right? 800 pages or something, right? So long, so big. Right? You don't need to make you don't need the instruction to make all the proteins. You only need the instruction to make some protein, right? 1 or 2 perhaps at this specific moment. So you make the copy of the segment that you need, and you do that through transcription. Okay? You get what's called a messenger RNA. That messenger RNA will leave the nucleus through the nuclear pores, and then it will go into the cytoplasm where the ribosomes will be waiting. Okay? So the ribosome will decode the information on the RNA, and then it will use it to make protein. So this process is called translation. Translation. Right? It's a different language now, right? RNA is nucleotides. What language is protein?
Amino acids.
Thank you, thank you. Okay, that is correct. The amino acid, right? That's why it's the translation, right? It's different language, right? Transcript means same language. You understand this is the central theme of biology, of how information flows. Starting at the DNA, we get the RNA, and then we get the protein. All right? Let's do some poll questions. Which of the following is a prokaryote? Remember, prokaryote is the noun, prokaryotic cell. That is the adjective, right? All right, let's launch the poll. Okay, anyone else before I close it? You know, that's not too bad. 78% of you who answer got it, right? Prokaryotes are like bacteria, right? And Piranha, I think. I'm sorry if I'm pronouncing it wrong. She said it was the E. coli, right? That's an example of bacteria. And that's what makes it a prokaryote. Okay, liver cell is in humans, right? Like that's eukaryotic cells. HeLa cell is from the ovarian cancer. That is human as well. Amoeba, also eukaryotes. How about this one? I must have told you 5-6 times already. I love to repeat myself. So hopefully you will remember the answer. Okay, looks like we're a little bit split here. The function of the nucleolus. The nucleolus produces ribosome, okay? Making a copy of DNA in the form of RNA, that's the process of transcription. It happens in the nucleus, but not like, that's not the function of the nucleolus, right? The nucleolus is like the seat on the inside, right? That's what makes the ribosome, all right? That's that. Okay, we're gonna move on to the endomembrane system next. We're almost there. I think we might even be done a little bit earlier than 2 o'clock. So let's look at the endomembrane system. This is a network of membranes that forms tubes and sacs within the cytoplasm. Okay, working together, they will help produce, process, and digest things like lipids and proteins. Okay, I'll tell you which part does what in a bit. It also helps recycles and dispose of molecules that are either no longer in need or in some cases worn out particles. Right, so that's the overall function of the endomembrane system. Like right now, you look at this like it might not make sense immediately, okay? But after we draw it out, I promise you it will be better. So here are the key components of the endomembrane system, and we actually already draw 3 of them, right? We have the nuclear envelope, which extends into the cytoplasm to form the rough and smooth ER. We have something called the Golgi body, sometimes also called the Golgi apparatus. And then we have the lysosome. So let's take a closer look at this. We already know the endoplasmic reticulum is an extension of the nuclear envelope, right? And then we have these rough ones with the ribosomes on it. So what exactly does it do? Now this part is hard to explain, okay? I will try my best here with some diagrams. I mean, you can draw along if you want, or like maybe it's better to just watch this part, okay? So I'm gonna show you the rough ER. Okay, the rough ER. What does it do? Okay. So I'm gonna zoom in on one of, you know, earlier I was drawing all these like tunnels, right? Okay, so I'm gonna zoom in on one of those. All right, so it's gonna look maybe something like this. I'm gonna zoom in on that. Okay, so this is the inside of the structure, and this is just the outside, right? Think of it like what's the best way of think about this? It's like, you know, those swimming noodles, those like long floaty styrofoam things that you can use at the swimming pool, right? That's kind of like that. Like this is the inside of it, okay? And then this is just the outside. So here is the ribosome sitting outside with the large and the small subunit, like the burger I talk about, right? And then you will have, let's say, this piece right here. That is the messenger RNA, right? A copy of the DNA that has the instruction to make the protein. That is our ribosome. And it's gonna make the protein, the amino acid, connect them together one by one as per the instruction of the messenger RNA. Okay? So afterwards, after it builds the protein will fold up to form a 3D structure. Remember, the sequence is just a primary structure, right? So it still has to fold up to form the helices, right? It's gonna, you know, come together to make a 3D shape, and now you have this protein. Okay, so this is protein, huh? Protein. This protein will move to the edge of the rough ER. And this is the part that's a little bit hard to show. I hope you understand the diagram. Usually I cannot show you animation in class because when I show you and then I upload the whole section on YouTube, like Google has this AI that detects material that is copyrighted, and then they will say, oh, your clip contains videos that is produced by someone else, and then they will ban my recording, right? So that's why I don't have an animation to show you. But you can find the animation in the video resource that I post on eCentennial, right? If you need to visualize this better. Okay, but let me try my best here. Okay, it's gonna look like this. Okay, so that protein will move towards the edge, right? And then it will pull a little bit of the membrane with it, and eventually it will pull so far out that it will just like pinch off. Can you visualize this? Give me some feedback here, guys. Let me know if any of this is making any sense.
I guess you could say in a way that it's like splitting away, right? Like
yeah, that's
becoming
exactly
its
what
own
happened.
entity kind of thing. Yeah.
That's right. It's like, you know, if you have some Play-Doh and then you just like pinch a ball off. That's one way of visualizing. Any question about what I'm trying to illustrate here?
Also, just to clarify, so the red scrunchy thing, the protein part, right? Is that like the beta sheets, alpha helix
Yeah,
thing?
all that stuff, right? And then it folds together to form a 3D ball, right? With the tertiary structure and whatnot, right?
Okay, yeah.
Okay, yeah. So in essence, the rough ER works with the ribosomes to produce protein and package it nicely into a bubble. This bubble is called a vesicle. All right. This is the professional drawing. You have the ribosome here, and then this is the rough ER, and you can see the polypeptide, right? Which will fold up into the protein it will push itself towards the edge. It will continue to push out until this snaps off, and then you have yourself a vesicle. Okay? A vesicle is just like a bubble, okay? A bubble with a protein inside. Okay. We will come back to smooth ER later on. I just want to continue the story about the bubble. What will happen to the bubble? Okay, let's go back to our picture here. And now you should keep drawing, all right? It turns out there is another organelle that's part of the endomembrane system. And this one is like pancakes, okay? It's like multiple layers stacked one on top of each other. Right? So this one here is called the Golgi body or Golgi apparatus. Okay, Golgi body or Golgi apparatus. It doesn't really matter. You can say either one of them. Okay, and it's a series of membrane. And the job of the Golgi body is to sort, modify, and repackage the content of vesicles from rough ER. Okay, copy that down first, and then I will explain to you what it means. So you see this bubble that pinches off from the rough ER earlier? Right, this is vesicle, right? Well, it turns out the protein is not quite ready for use yet. Okay, there might be some extra modifications that you need to make to it. You might need to give it like a special tag so that it knows where to go. Right, so it will need to travel to the Golgi body. And the Golgi body is like the sorting center for packages. Okay, like the what do you call that? The post office. Okay, they receive all these packages. It needs to sort them. It needs to know what goes where. And sometimes, you know, if the package is broken, they have to fix it and rewrap it. Right? That's essentially what the Golgi body does. So the vesicle will come and fuse with the Golgi body. It will get passed forward and sometimes backwards a few times, so that they could sort the content, they could make the necessary modifications, like add a tag to it, like I said, like literally add a tag to it so that it knows where to go in the cell, repackage it, right? So at the end, it's gonna come out as a brand new vesicle, okay? And the content is now modified, okay? I'm just gonna use a different color. Right, so that is a repackaged vesicle. It's still protein inside, okay? It's just like got a little bit repackaged, okay? It's revamped. It's better now. It's ready for use, essentially. Okay? Sorry, I'm like moving a lot. My back is killing me. Ugh. All right, so what do we do here? The vesicle has several destinations, okay? Number one, it might be ready to be released from the cell, okay? So the protein will be released from the cell immediately, okay? So remember the cell membrane that I erased? Let me just bring it back for a second. Okay, so that's the cell membrane. So this vesicle can travel all the way from here all the way to the cell membrane, and then it will fuse with the cell membrane, and then the protein that's in there will leave the cell. Okay, maybe it's a hormone, maybe it's an insulin. It goes into the bloodstream. We need it right now. Okay, so that's one possible function. Okay, a second function is that this is harder to understand because we haven't talked about it before. It is going to become part of the cell membrane. Okay, we know the cell membrane is made up of phospholipids, a little bit of cholesterol, but it turns out there are quite a bit of proteins on it as well. All right, we'll learn more about it next lecture. So yeah, you might have a protein right here as part of the cell membrane. Or maybe you don't need it right away. Maybe you just make them so that you have a good stock, and in case you need it, you will have them ready, right? So it can be stored in the cytoplasm until it's needed, right? It's hard to make things on demand in the body. Sometimes things happens really quickly, right? So rather than making them on demand, you have them stored, and then you can release it all at once when you need them. Okay. Finally, the last option is that it will become what color should I use? I'm running out of colors here. There's so many things. Becomes something called lysosome, which I'll explain. Okay, so these are different paths that the vesicle can become. Okay. Questions? Okay, almost there. Like I said, we'll come back to the smooth ER later on. This is the Golgi body, right? You see is multiple layers of membrane stacked together, and the job is just to store, repackage, sort, repackage, and tag them, as in modify them, put a sticker on them so they know where to go. Where will the protein goes from the Golgi body? I just roll it down for you, right, with all those arrows. So you can see here is a vesicle coming in, arriving at the Golgi body. It will fuse with it. It will go back and forth, and then you get repackaged. Sarah said, okay, yeah, sure, here. I'll post this as well. Probably not today, okay? Like we still have to add more things to this note, but I'll post it when we finish the whole lecture 3. But here it is, okay? Take a screenshot if you need to. Okay, great. Where was I? Right here. All right. So one of those options, right? We have 4 options. One of them is that it will form the lysosome. A lysosome. What is a lysosome? Lysosome is part of the endomembrane system, and it contains a mixture of digestive enzymes. You know about enzymes already, right? They are responsible for breaking things down. They catalyze, they speed up chemical reaction, right? So the enzymes that is in the lysosome is responsible for breaking down macromolecules, okay? They are also responsible for recycling old organelles, right? Let's say we have an organelle that's damaged. It's no longer performing its function, then it makes sense to get rid of it, right? So that it doesn't cause malfunctioning of the cell. So that organelle will fuse with the lysosome, and then it would be digested, and then we can recycle some of the locked up nutrients. Sometimes your white blood cells, for example, white blood cells, they will eat up a bacteria. Okay, so I know it says food here, but let's change it to bacteria so it makes more sense. I mean, it could be food as well, but let's just change it to bacteria, and then after the cell eats the bacteria, you will have to break it down. Okay, and so the lysosome will come, breaks it down, digest the bacteria, and you do this many times until you've cleared the infection, right? So that's the That's the function of lysosomes. Okay, lysosomes. It also has an additional function. Sometimes we need to deliberately kill off cells in the body. That is called apoptosis. Okay, programmed cell death. Okay, you purposely kill off the cells, either because you no longer need them, or the cell is too old, too worn out, and it's kind of wonky. It's like, you know, not doing its job properly. It's causing problems. Then you want to kill them off. All right. So lysosome, having all these enzymes in it, can release all the enzymes into the cytoplasm, and those enzymes will essentially kill the cell off. Right? That's apoptosis. Did you know at one point during our embryonic development, we have webs between our fingers? Okay? Clearly, we don't have webs anymore, thank goodness, right? But those the cells in the web, needs to be killed off as part of our normal development, right? So how do you kill those cells? Well, you need to trigger apoptosis, right? Apoptosis, programmed cell death. Does that make sense? So think of the lysosome as the recycling depot, right? Breaking things down to unlock the valuable resources. You can also think of it as the garbage disposal center. If there's a bacteria coming in, you want to dispose of it, kill it, right? And it's also capable of killing the cell when the situation arises. Okay, we call that apoptosis. There are a group of diseases called lysosomal storage disease. These are hereditary diseases. That means they can be passed on from our genes. Because the enzymes in the lysosomes are made up of proteins, right? Enzymes are proteins. You know that by now. And so the instruction to make them is in the DNA. If the DNA mutates, you might not make the enzyme anymore, right? So depending on which of those enzymes you're missing, right, the result would be a particular kind of lysosomal storage disease. So in Pompe disease, there is a mutation that causes the enzyme to be missing, and you cannot process glycogen anymore. So there is a harmful buildup, accumulation, it's built up, right, of glycogen in muscle cells. Right? And that would be a particular problem if it's affecting the heart muscles. Right? It's interfering with how your heart beats, right? So that could be potentially lethal. There's another one called Tay-Sachs disease. In Tay-Sachs disease, the enzyme that is responsible for breaking down lipid is missing in the lysosome, that is, right? And then there is excess buildup of lipids in nerve cells, which can interfere with brain development. Okay, so these are just some examples. There's a whole list of them, right? Again, depending on which enzyme is missing, right? The condition is going to manifest differently. And that's all I wanted to do today. I know I haven't do the smooth ER yet. I will pick that up next time. But right now it shows a good story, right? Okay, we have the protein being made in the rough ER, the vesicle that are budding off from it, going to the Golgi body, right? Gets repackaged, sorted out, and then you have a new vesicle that might be released. They might stay in the cytoplasm. They might become part of the protein, or they might become the lysosome, right? Which does the thing I just told you a few minutes ago. So that's it. Again, thank you for coming, and good luck with your bio quiz. I will try to mark it as quickly as I can. But yeah, just do them before 11:59 tonight. Okay. Have a wonderful rest of your day. Bye for now.
Mr. Wong.
Bye-bye.
Thank you.